Good evening, students. So this is our today is our fourth class of biochemistry, Alhamdulillah, and we have finished with the general overview of vitamins from first aid as well and from Lippincott as well. Okay, and then we started with vitamin A, vitamin B1, vitamin B2, and B3. Yesterday, we did vitamin B2 and B3 in a lot detail, okay? And our lecture was of 1 hour 30 minutes yesterday. So today we will be studying vitamin B5 and B6. Now, this topic looks like not even half page of biochemistry, but the problem with students is they have really bad basics of biochemistry and they don't really understand what it is written when they read this book. So my focus is to let you understand at the level of the textbook Lippincott, okay, and explain you every little word that is written here, okay. So let's begin our lecture from vitamin B5 today. Bismillahir Rahmanir Rahim. So the name of vitamin B5, first of all, vitamin B5 is a water soluble vitamin. Okay, we have 4 fat soluble vitamins, that is A, D, E, and K, and vitamin B and C both. All vitamin B, either it is B1, B2, B3, B5, B6, B7, B9, or B12. Okay, all of them are under water soluble vitamins, and vitamin C is also a water soluble vitamin. Okay, so the name of vitamin B5 is important. I told you in my first lecture that all of these names you have to remember because they don't ask MCQs ever with these symbols. Okay, they will not ask you like B5, but they will use this name pantothenic acid. So you should know the names of vitamins really well. Now, what does vitamin B5 do? You see, it is an essential component of coenzyme A. Now, in my last previous lectures, I have been repeatedly telling you what is a coenzyme. Okay, that is an enzyme that there is a main enzyme that converts the substrate into a product. Now, where's my pen? Okay, now this is the are we recording our lecture? Yes. Okay. This is a substrate, for example. Okay. And now this is the reaction and this is the product. Okay. So what is going on over here is there is an enzyme that catalyzes this process. Okay, now it depends on the name of enzyme. What does it do? For example, if it is a dehydrogenase enzyme, it will remove one hydrogen. Okay, if it is a kinase enzyme, it will phosphorylate, that is, it will add put a phosphate group, PO3 with a negative charge. Okay, it will add a phosphate group. If it is, for example, hydrogenase, okay, or if it is a dephosphorylase, it will remove the phosphate group. If it is a hydrogenase, it will add a hydrogen group. So these are the enzymes. That's just I'm telling you an example. So they are the main enzymes. Now, what is a coenzyme? What is a coenzyme? Co means a product or a let's take it as a person. Okay, let's take enzyme is one person and coenzyme is another person. Okay, so a coenzyme is that person that is helping this enzyme to function. For example, in certain situations we see that there is this when, like in previous lectures when we were studying this synthesis of acetylcholine, in pharmacology we studied, in physiology we studied. So what we studied in that there is a choline, okay, there is a choline that's coming, and that choline is converted to ACH, okay, and there is an enzyme called acetyl choline transferase that is transferring acetylcholine transferase, okay, that is transferring this choline to form acetylcholine. But what is actually happening is, you see, there is this acetyl CoA, okay, there is this acetyl CoA, acetyl coenzyme A, that comes and helps this acetylcholine transferase to convert choline into acetylcholine. We have, like, in our pharmacology or in your physiology lecture, we studied this mechanism again and again, right? If I want to show you that thing, I kind of wait a second. Just wait a second. Okay, here I found it. Just wait a second, people. Okay, so resume share, and let me clear this all drawings. Now this was our this what we were studying today. We were studying this in pharmacology, and I was kind of giving you the example of this right now, that what a coenzyme does. Now you see, this was choline, okay, and then it converts into acetylcholine. So this choline is a substrate. This is a substrate, and this acetylcholine is a product. Now substrate is converting into product, okay, via an enzyme called choline acyl transferase. That is, it is transferring this choline, and this acetyl CoA right now over here, AC for acetyl and Co is for coenzyme A, okay, this acetyl CoA is working as a coenzyme. It is a coenzyme. This choline acetyl transferase is an enzyme, okay, so enzyme is the main thing that is doing its work, and the coenzyme is the enzyme that is helping enzyme to perform its action completely, okay, so this is the difference. Now what happens is, you see, the essential component of coenzyme A is vitamin B5, and if it is an essential component of coenzyme A, at what particular levels it must be helping at CoA level, that is, you can see in TCA cycle, when acetyl CoA when pyruvate from the product of glycolysis, pyruvate is converted to acetyl CoA. CoA enters here at this level, so it requires vitamin B5. Okay, it requires vitamin B5 to perform this action. Then it is also a cofactor for acyl transfers. Now, acyl transfers are, you will see this in fatty acid synthesis and fatty acid degradation, okay? And fatty acid synthase is another enzyme. So it is a component of coenzyme A, fatty acid synthase. Now, let me show you a diagram, a picture of, first let me check this page in my textbook, like in my first aid. What is the page, so that you can go quickly to that page in your book. Okay, people, turn to page number 2. 89 in your first aid book. Okay, let me take you to page number 89 in this book. Okay, let's just quickly go to page number 89. I will pause the recording. Okay, now here we have this page number 89, and what I want to show you is, you see, this is the process of fatty acid synthesis and degradation. And at this particular level, you see fatty acyl CoA written over here. You see this fatty acyl CoA dehydrogenase enzyme present over here. Okay, so this vitamin B5, this is my, and this is another book, a PDF. Okay, so it is my first aid book, so we can study it from here as well. You see, it's just a bit blurred, but rather than going back up, I can open this one for you, and you can check it in your book as well. Okay, so we said that it is an essential component of coenzyme A and fatty acid synthesis. And when we're talking about coenzyme A, we are talking about CoA and a cofactor for acyl transferase. So now you see, at this particular level, this is as fatty acyl CoA, and you see acyl CoA is adding up over here. You see this fatty acid is combining with CoA at this level, okay, and then it is coming at this level, and then it is going to the and it's becoming the acetyl CoA and entering into the TCA cycle, and also forming the ketone bodies. So these mechanisms, we require vitamin B5, pantothenic acid. Okay, another point where we require this vitamin B5 is we require it in the enzyme called fatty acyl CoA synthetase, or you can say synthase. Okay, so this is the enzyme in which you require this vitamin B5. Now, what will happen if a patient will be deficient of vitamin B5? Now, you understand that like till now we have already been studying since a long time that all these vitamins are essential for, you know, for our hairs, for our nails, some for our sight, some for something, but we never noticed that these vitamins, especially the B complex, is so much essential for the metabolisms going inside our body. That is carbohydrate, protein, and fatty acid metabolisms. We study these enzymes as cofactors and coenzymes in so many reactions, that if these enzymes will be deficient, what will happen to our metabolic activities? Okay, so just for in case, if this vitamin B5 is not present in your body, what will happen? This process will not go on. Okay, and when this process will not go on, you will see we need this step. This is a very important step that we call carnitine shuttle. Okay, this is cell cytoplasm. This is cell cytoplasm, and this is mitochondria. Okay, so from cytoplasm of the cell to the mitochondria of the cell, this process goes on, and in the and this is this this cell is of liver. Okay, this is a hepatocyte. So all this fatty acid metabolism undergoes in a liver cell, that is hepatocyte. Okay, so in the mitochondria of hepatocyte, this whole process goes on, and this TCA cycle also goes on and over there. Now, what happens when we have this deficiency of vitamin B5? It will not be giving in this CoA as a cofactor to these enzymes, and we will not be forming fatty acyl CoA. When we do not have this fatty acyl CoA, and when there is a decreased ability to break down this fatty acids, okay, into this acetyl CoA, this fatty acyl CoA, will be accumulated in the cell cytoplasm. Okay, or if it is reached up here, it's even at this particular step, this will be accumulated in cell cytoplasm, And when this will be accumulated in the cell cytoplasm, the patient will get deficiency of glucose. Why I'm saying glucose, why the patient will get deficiency of glucose? The reason is because TCA cycle gives glucose. It is a carbohydrate metabolism. We study TCA and carbohydrate metabolism, right? So the patient will be deficient of glucose, that the patient will show symptoms of hypoglycemia. Okay, similarly the patient will not be forming ketone bodies, so the patient will be in hypoketotic hypoglycemia. Okay, and this is such a dangerous condition. It leads to hypovolemic shock. Okay, it leads to hypovolemic shock, and the patient can like undergo into coma as well. The patient's liver can get dysfunction. So if these are certain things that are also, that can also be some processes that can also be hereditary, or there can be problem in vitamin B5 intake, or those children and infants who belong to a very poor background, and they do not get all this good stuff to eat, which are full of vitamins. So they get deficiency of vitamins, and we see all these problems going on into them. They're very lethargic, and they have this liver dysfunction, which can lead to cirrhosis, and they have ascites, and all these functions in these tiny little children. So this vitamin B5 is very important to study. Okay, so let's go back up. Let me pause the recording. Okay, so so you understand the functions of vitamin B5, that the essential component, it is an essential component of coenzyme A and fatty acid synthase. So you have to remember these names of enzymes along with this, okay? Because in all the functions we have been studying, for example, in vitamin B1, when we were studying, I told you that B1 is an important component of dehydrogenases enzymes, okay? And when we were studying vitamin B2, I told you it is an important cofactor for redox reactions. Okay, also B3 was an important factor for redox reactions. Similarly, B5 is important component of coenzyme A and fatty acid synthase. Okay, and then likewise, when we'll move on B6, B7, for B7 for carboxylases. So it's like you have to learn the core name of this enzyme, in which they are important, because this can be asked in your MCQs. Now let's study deficiency. One common thing that I told you regarding the vitamin B complex deficiencies were three things, okay, and that were diarrhea, dementia, and dermatitis. I told you to remember 3 Ds. Okay, yesterday somebody was like texting me, and when I was delivering the lecture, he said that we have 4 Ds, and the 4th D is death. So definitely the patient will undergo death ultimately when he or she will be suffering from these B complex deficiencies, because they are playing so important role in the metabolism. When the patient will not have enough glucose to perform the basic activities, the most of the glucose is used by brain. So it's the organ that use the most glucose. So when you will be deficient of glucose, lots of things will be compromised in your body. So it's not a joke to have a vitamin B complex deficiency. So that is an ultimate point. But nobody asks that in the MCQ. So you have to kind of remember 3 Ds. That is diarrhea, okay, diarrhea. That is dementia. Dementia means forgetfulness, okay, and dermatitis. So remember these 1, 2, and 3 Ds. Then you see enteritis. Enteritis means inflammation of your gut lining, okay. Your gut lining will be inflamed. Alopecia means baldness, okay, and adrenal insufficiencies. So you will see all these problems in a patient with vitamin B5. Okay, this is really important to remember these things. Okay, so I really don't know why we're having adrenal insufficiency, so you can like kind of Google it and search it, but the other things are really obvious. Okay, because it's hindering the processes of metabolisms. Okay, so next is we have vitamin B6. Okay, so the vitamin B6 is pyridoxine. Vitamin B6 is pyridoxine. They don't ask much MCQs on B5. On the contrary, B6 is really important. So the important vitamins in B complex asked in MCQs are B1, B3, B6, B9, and B12. Okay, so you have to focus on these vitamins more. So that is why I will be paying more time and more attention on these vitamins. I explained you in the whole one lecture B1, and we spend most of our time on B3. So today our main vitamin is B6. Okay, so let's study vitamin B6 together. So vitamin B6 is also called pyridoxine, okay, or you will heard it pyridoxine phosphate. So if you see both of the names, both of them are correct. Okay, so what is its function? It is converted to pyridoxal phosphate, which we call as PLP. Okay, pyridoxal phosphate is a cofactor which is used in transamination, example ALT and AST. Okay, now it is very important to know this point that ALT and AST are the liver enzymes. Whenever you ask a patient to go and check his liver profile, there is a test which is called as liver profile. Okay, um, so this liver profile has this certain enzymes. What is going on? Liver profile has this certain enzymes called let me tell you what. Number one is this ALT. Okay, number two is AST. Number three is GGT. Okay, number four is you have what I can say, you have this in some cases you got this HDL and HDL and LDL also here, but the main specific enzymes are these ALT, AST, and GGT. Okay, now one more important thing to remember is we use a word called SGOT and SGPT. SGOT and SGPT in case of ALT and AST. Okay, so also remember these two alternative to these two names. So these are called alanine transferase and aspartate transferase. Okay, AL stands for alanine and AS stands for aspartate. They both are proteins, amino acids. Okay, alanine and aspartate are the amino acids. So this is called transamination. Okay, and these enzymes are liver enzymes. So this vitamin B6, okay, it is converted to this pyridoxal phosphate, that is PLP, okay, which is a cofactor for these enzymes. So if you have deficiency of vitamin B6, you will see deficiency of these two as well. Okay, so this is important. This is really important to, so the key word which you have to remember is transamination. Okay, you have to remember transamination along with ALT and AST. Now, the other enzymes in which it's helping is this decarboxylation reactions, or you can say decarboxylase enzymes. Okay, and then you see glycogen phosphorylase. So it is helping in glycogen phosphorylase. It is helping in decarboxylation enzymes. Now we have this. They have this when you'll be studying, because we are studying right now vitamins. So when you will be studying this metabolism, okay, so on page number 73 of your book of your first aid, on page number 73, there is an enzyme, or directly turn to page number. Let's turn to page number. Let me see in my book. It is, I guess, page number 74. Okay. So on page number 74, there is this mechanisms, and we have this glycogenolysis mechanism here. Okay. The breakdown of glycogen. They've not mentioned the enzyme name over here, but I can kind of tell you, like even in the book I can like kind of tell you. So let's go to that page. It's page number 74 and 73. Okay, so first let's go to page number 74. See this glycogenolysis. Okay, so let me, this is a very interesting page. If you will like kind of learn this page or understand these pathways, you will never forget. In Lippincott, you have a similar page, like page number 92, I remember. So you have to open that page also to understand these processes if you want in detail, because in first aid it's not that in detail. Now you see this is glycolysis, and in glycolysis you see this whole pathway is going on, and when we go from this glucose 6 phosphate to glucose 1 phosphate and UDP glucose, and then glycogen is formed. But then in the similar way, this glycogen can be degraded into glucose 1 phosphate, and from 1 phosphate to glucose 6 phosphate. So this process of glycogenolysis, that is breakdown of glycogen, over here in these steps, there is an enzyme which is called glycogen phosphorylase. Now you see 1, 2, 3, 4, they have not mentioned any number over here. So they have not mentioned glycogen phosphorylase enzyme name over here. But you have to remember and you have to understand that glycogen phosphorylase is the enzyme that is used in the process, in the steps of breakdown of glycogen. Okay, so let's go to page number 73. This is the chart which you need to mug. Literally you need to mug, because these are the rate limiting enzymes, rate determining or rate limiting enzymes, and they're asked straightforward in MCQs. So you see you have this glycogenolysis, and its rate limiting enzyme is glycogen phosphorylase. So this is the enzyme which can exactly stop the breakdown of glycogen. It is a rate limiting enzyme, and this enzyme use vitamin B6. So how important is vitamin B6? It is stopping the the process of the breakdown of glycogen. When your body is in fasting condition and you do not have enough glucose in your blood by food, your body undergoes the breakdown of glycogen into glucose. Then this glycogenolysis is converted to glycolysis, and then this glycolysis is converted to TCA cycle. Okay, so when your body is in fasting and you are deficient of vitamin B6 also, you this you your glycogenolysis mechanism will stop and you will kind of go hypoglycemic really bad. So this is the enzyme which is stopping this pathway, and it uses B6. Okay, so this is the concept you need to clear at this particular level. Now the other, I'm not going up back again. Let's see in this book. So we studied transaminases like ALT and AST. Then we studied decarboxylation reactions or decarboxylase enzymes. Okay, and then glycogen phosphorylase. So I kind of told you about the glycogen phosphorylase. Okay, I kind of told you about this. Now we have these decarboxylation reactions as well, okay, in which we remove the carbons and convert them to some other forms. Let me show you an example of decarboxylase reactions. But as in a whole soul, they also cause a problem to, they also cause problem in the metabolic reactions. Okay, so decarboxylase now you see this is a one example. This is one example. Pyruvate is converted to acetaldehyde and then ethanol. You know, ethanol is very important for our body, although it is like alcohol, but why it is important, let me show you. There is another picture in your book on page number 72. I guess we're on page number 73, so let's go on page number 72. So on page number 72, you have this, wait a minute. You have this ethanol. So you see that this acetaldehyde is converted, ethanol is converted to acetaldehyde and acetaldehyde into ethanol. So like vice versa. So in this picture, you see also acetaldehyde is converting to ethanol, but you have this pyruvate which is converting to acetaldehyde as well. And this pyruvate is the final product of glycolysis cycle. So pyruvate can be converted to this, and this can be converted to this and further. So what it will helping you in, it is helping you in this ethanol metabolism, which is really important. Because if you will not have this, ethanol metabolism going into your body, you will be like a kind of having this hangover symptoms. For example, like when there are people who drink alcohol a lot, because ethanol is an alcohol. Like we Muslims do not do, definitely, but there are other people in the world who drink alcohol, and that alcohol needs to be metabolized. Alcohol needs to be metabolized. So ethanol metabolism is important, and this acetaldehyde is somewhat coming from not, this is an opposite arrow. This acetaldehyde is also coming from pyruvate. Okay, from this pyruvate. Here it is. This pyruvate directly. This pyruvate is the end product of glycolysis cycle, because they have like, this is glycolysis cycle. When it is going downwards from glucose to pyruvate, it is glycolysis. But when it's coming from PEP to glucose, it is gluconeogenesis. Okay, do not confuse. Let's go to page number 74. It is very good picture over here. So you see, we call it glycolysis when it goes from glucose to pyruvate. But when it comes from phosphoenol pyruvate, that is PEP, to glucose, it is called gluconeogenesis. Means formation of glucose. Gluco is like glucose, genesis is formation, and neo, N-E-O, neo, means from other processes, like from proteins, from fats. So it is not necessary to produce glucose from glycogen. When we produce it from glycogen, we call it glycogenolysis. But when we produce this glucose, when we form this glucose from lipid metabolism, or when we form this glucose from clear all my drawings, when it forms from protein metabolism, so you see this whole process is like going on succinyl CoA, and then malate oxaloacetate, and then oxaloacetate is converting to PEP. Okay, and then this PEP will go straight up and will convert to glucose. So this, we can form glucose from protein metabolism as well. We can form glucose from lipid metabolism as well. So you see, we have this this little body, which is glycerol, and this glycerol, or these TAGs, or these fatty acids, they can be go to this step, and from this particular step, DHAP, it can from this step, it can enter into glycogenolysis, and then it can form glucose. So we have lots of pathways. So this neo word means from other substances like lipids or like proteins, okay, but not from carbohydrate. Fructose is carbohydrate, galactose is a carbohydrate. So not from them, but from lipid and from proteins. So the purpose of explaining this all process to you is to let you understand that this pyruvate can be converted to ethanol, okay? And ethanol is converted to acetaldehyde via an enzyme called carboxylase. And that enzyme helps in this ethanol metabolism. Okay, again, I'll show you this picture. So this whole process. So vitamin B6 is really important in all these mechanisms, okay? So I guess now we are clear with transamination, decarboxylation reactions, and glycogen phosphorylase as well. Now let's move further. Synthesis of glutathione. Yeah, now this is really important. Synthesis of glutathione, cystathione, heme, niacin, histamine, neurotransmitters, including serotonin, epinephrine, norepinephrine, dopamine, and GABA. So see how many mechanisms this B6 is important. If you will not have B6, you will not be making these neurotransmitters. So no sympathetic reactions, no parasympathetic reactions, no reactions in your renal vasculature, because dopamine is important. You will not have the happy and the sad hormone, that is serotonin or dopamine. You will be lacking GABA, okay, for many of your brain reactions going on in your head, because GABA is the main neurotransmitter, inhibitory one, that is responsible for that chlorine influx, okay, and you will not be having histamine as well, because histamine is helping you in your what can I say, in your respiratory processes, okay. Then you will be lacking vitamin B3 also. So if you remember, we kind of studied that for the production of vitamin B3 we should have B6 and B2. So I told you yesterday. So you see, niacin is B3, and it is helping in the formation of B3 as well. So no B6, no B3. The patient will also comes with pellagra. So you have to remember this point as well. Then let me explain you about glutathione and cystathione more. So this book is a complete book. It has all those things that you need to remember. Let me pause and search first. Okay, so now you see, this is this pathway. We have this glutathiones. Okay, so you see glutathione reductase and glutathione peroxidase. So this is a glucose 6 phosphate dehydrogenase deficiency, and we see glucose 6 phosphate dehydrogenase in your steps to glycolysis and in your steps to glyco gluconeogenesis. Okay, glucose 6 phosphate is an intermediate step. So you see glucose 6 phosphate dehydrogenase, it is using this NADPH and NADP, which is coming from electron transport chain into and it is using this glutathione reductase. So you see, these mechanisms are so important. Now glucose was converted to glucose 6 phosphate. Glucose 6 phosphate is using this enzyme glucose 6 phosphate dehydrogenase. Then this NADP is coming from electron transport chain, ETC, okay, and electron transport chain. Now these two things are combining together using this enzyme. It is producing this product, and also as NADP is converting to NADPH. Then what's happening, this NADPH is using glutathione reductase along with this GSSG in oxidized form, and they are converting this into this reduced form. So this whole process has involved glycolysis and electron transport chain both. Okay, so the importance of this is to keep this enzyme. The importance of these enzymes is to keep this glutathione into the reduced form, okay. Because if you will like study pathology, journal pathology with me, I will teach